Assalamualaikum warahmatullahi wabarakatuh. My name is Anita Hana and today I will be presenting about my industrial training at Forest Research Institute Malaysia, Frim. Assalamualaikum warahmatullahi Forest Research Institute Malaysia, also known as Frim, is one of the leading institutions in tropical forestry research in the world. The institute sits adjacent to the Bukit Lagong Forest Reserve in the Kepong, and Frim is working toward attaining the recognition as a UNESCO World Heritage Site. Its nomination to be included into the tentative list of the UNESCO World Heritage Sites was accepted at the 41 World Heritage Committee's meeting. This institute is led by Datuk Dr. Abdul Latif Mohamad as its Director General. So during my industrial training, I was assigned in herbarium department. As an internationally recognized herbarium, it houses about 300,000 specimens with over 770 types. So my main project during industrial training at Frim is documentation on bryophyte in Forest Research Institute Malaysia, Frim itself. The first task for my project is to attend a talk by one of my supervisor, Chia Yi Hong, and he talked about the introduction of bryophyte. He teach us how to collect bryophytes in the field and how to identify the species of collected bryophytes for further research. The next task is preparation for fieldwork. Preparation for fieldwork has many aspects need to be revised before we actually collect the samples in the field. The most important thing is to prepare a lot of envelopes out of recycled A4 because we need to consider the cost. That's why we use the recycled A4. So the purpose of preparing envelopes is to store the bryophyte sample after we collect it in the field. So the next step is fieldwork. Me and one of my supervisors, Chia Ying Hong, done the general sampling along the Sebasar Trail and the Sungai Keroh waterfall. There is two types of sampling for bryophyte in the field. One is quadrate sampling and the other one is general sampling. My supervisor decided to do general sampling because it's less time consuming and it less budget. The next task is preserving the bryophyte sample in the field. I have to store the collected bryophyte sample from the field inside the freezer with 80 degrees for 7 days. The purpose for preserving the bryophyte sample in the freezer is we need to eliminate any type of live specimen from the surface of the bryophyte or inside the bryophyte, such as tiny insect or worms. It is because we are forbidden to bring any live specimen inside the herbarium at Frim. One of the reasons why they forbid us from bringing the live specimen inside the herbarium is to protect the older specimen from rotten or destroyed by the fungus or the insects. So the next task is grouping bryophyte specimens. As we know, bryophyte have three different groups, which are mosses, liverworts and hornworts. So grouping each samples can make it easier for us to proceed with identification of species later on. So first we need to take out the Ziploc bag from the freezer and place it in herbarium with 23 degrees Celsius to 24 degrees Celsius for 24 hours. So each envelope placed under the stereo microscope, and with the help of light extension, the bryophyte sample was observed under the microscope. The bryophyte in the envelopes carefully examined, and the group of bryophytes were determined. So the next task is identifying species of the selected bryophyte samples. So my supervisors choose 10 samples from the bryophyte samples collected in the field and brought it to University of Malaya to examine and observe so that we can identify the species of the bryophytes. There is few steps that need to be followed to identifying the species. First, we need to place the selected sample under the stereo microscope. Next, for group mosses, the leaves were dissected and placed inside a petri dish containing water. But it's slightly different for group liverwort. The parts of the liverwort were picked and placed inside the petri dish containing water. The slide will observe under stereo microscope. With the 8 forceps, the dissect leaves were placed on both sides, ventral and dorsal, which is the back side and the in front side. Then the slide were observed under the compound microscope, and the species were determined by referring to the prologue of the groups, mosses and liverwort. Unfortunately, the next task is write an article about the bryophyte sample. The next task is write an article about the bryophyte samples. My supervisor picked 4 out of 7 species that we have identified at University of Malaya before. The selected species were Hyophila involuta, Legionella tuberculosa, Leptolegionella elliptica, and Calipogaya arguta. So in summary, the scope of research in the field of botany were determined by doing tasks in fieldwork. The technique of processing, management herbarium and maintenance of plant specimen were observed by doing some tasks such as pressing, mounting, sewing and shelving. The techniques should be done in sequence to avoid any inconvenience among the staff. So my recommendation is internship students should experience more fieldwork to gain additional skill that help them for the further research. In addition, the internship students should attend some courses regarding the management of herbarium to provide them with proper knowledge. So that's all for me and thank you so much for watching. Assalamualaikum warahmatullahi wabarakatuh. Bye.